Welcome to the Explainer. So, today we're diving into something that is literally keeping you alive as you hear this. Think of your cell membrane as a hypersecure, bustling microscopic border control. It's the ultimate gatekeeper, deciding exactly what gets the VIP treatment to nourish the cell and what gets kicked out as waste.
Okay, let's look at the roadmap for today. 1. The border control setup. 2. Passive transport, which is basically free passage. 3. Active transport, or pumping uphill. 4. Bulk transport for the massive cargo. And 5. A quick summary to wrap it all up.
Let's jump right into Section 1, the border control setup, specifically permeable membranes.
So before we get into how things move, we really need to understand the wall they're moving through. A cell's survival completely depends on maintaining this selectively permeable barrier. It's not a solid brick wall, but, it's definitely not a wide-open door either. Down at the molecular level, this is your lipid bilayer. Now, if you're a really small or uncharged hydrophobic molecule, you get that VIP treatment. You can slip right through the lipids without any help at all. But if you're a larger molecule, or if you're carrying an electrical charge, blocked at the border, you're going to need special clearance to get across.
Which brings us to Section 2, passive transport. Free passage. Passive transport is all about going with the flow, and the best part, it doesn't cost the cell a single drop of energy, no ATP required whatsoever. It relies entirely on natural concentration gradients. It's kind of like a crowded room. People naturally just want to spread out to where there's more space, right? Molecules do the exact same thing, moving from an area of high concentration down to an area of lower concentration. And this free ride basically comes in 3 distinct flavors: simple diffusion, facilitated diffusion, and osmosis. Let's look at simple diffusion first. Molecules just glide effortlessly right through the membrane. They start in a highly crowded area and just naturally drift down into the less crowded space inside the cell. It's spontaneous, it's incredibly steady, and again, absolutely zero cellular energy is needed. To put that into perspective for a second, just take a deep breath. Right now, oxygen just filled the tiny air sacs in your lungs, the alveoli. Because the oxygen concentration is super high in that air sac and very low in the blood cell passing by in the capillary, that oxygen just diffuses straight across the membranes, right into your bloodstream. No energy required. This simple diffusion is quite literally actively keeping you alive this very second.
But what about those blocked molecules we talked about? The ones too big or too charged to just slip through? Well, enter facilitated diffusion. Your cells have transport proteins embedded in the membrane that act as dedicated border gates. Channel proteins, for instance, are basically open tunnels for specific ions. Meanwhile, carrier proteins actually grab the solute, physically change their own shape, and drop it on the other side. Now, they're still moving from high to low concentration, so it is still a totally free passive transport. They just need a little structural help to bypass the lipid bilayer.
Okay, but this raises a pretty big question. What happens when the cell needs to stockpile nutrients? Like, what if it needs to force molecules from an area where there are barely any into an area that's already jam-packed? How do you force things to go against their natural flow?
The answer to that is active transport. Unlike passive transport, where things just lazily float downstream, active transport is the really hard work of pushing cargo uphill. You're moving from a lower concentration to a higher concentration, actively fighting that natural gradient. And just like pushing a heavy boulder up a hill, this requires energy. In the cellular world, that energy currency is known as ATP.
Let's explore that in Section 3. Active transport, pumping uphill. What's really fascinating here is looking at the blueprint of the microscopic machinery fighting those gradients every single day. This is the sodium-potassium pump, absolutely vital to your survival. It's constantly working, especially in your nerve cells, tirelessly pumping sodium out of the cell and potassium into the cell, totally against their natural gradients. Let's break down this mechanical marvel step by step. It happens in a precise 5-step dance. Step 1. Inside the cell, 3 sodium ions bind to the protein pump. Step 2 is where the magic really happens. A molecule of ATP swoops in and phosphorylates the protein, which just means it attaches a phosphate group. Step 3, this burst of energy literally forces the pump to change shape, throwing those 3 sodium ions right out of the cell. Step 4, with this new shape, the pump now perfectly fits 2 potassium ions from the outside. They bind to it, and that triggers the release of the phosphate group. Finally, step 5, Without that phosphate, the pump snaps right back to its original shape, dropping the 2 potassium ions inside. And then, the whole cycle repeats. The real secret sauce to this entire operation is phosphorylation. The mere transfer of that single phosphate group actually changes the physical shape of the protein. When it shifts shape, it completely loses its grip on sodium, tossing it outside, while suddenly gaining a really high affinity to grab onto potassium. It's an incredibly elegant, microscopic shape-shifting machine running entirely on your cell's built-in batteries. Moving on to Section 4, Bulk Transport, Massive Cargo. So, what if you need to move something truly massive? I mean, stuff way too big to fit through a tiny channel or a molecular pump. When materials are just too bulky, think whole microorganisms or giant fluid droplets, the cell membrane actually has to physically fold and reshape itself to handle the shipment. It swallows things in, which we call endocytosis, or it spits things out, which is exocytosis. And because it's moving huge sections of the actual membrane to form these little bubble-like carrying cases called vesicles, it uses a massive amount of ATP energy to pull this off. Let's zoom in on endocytosis for a second, bringing things inside the cell. It generally comes in 2 forms that are actually super easy to remember: phagocytosis, which translates roughly to cellular eating, and pinocytosis, which is cellular drinking. You can really see the contrast in how these function. In phagocytosis, the cell membrane actually reaches out with extensions called pseudopodia to grab and engulf a massive solid particle, creating a large food vacuole. It is literally eating it. On the flip side, with pinocytosis, the membrane gently dips inward. It just pinches off tiny, fluid-filled droplets of dissolved solutes to bring them inside. It's just taking little sips from the surrounding environment. Just to totally lock this in for you, here's a clear breakdown. Phagocytosis handles large, solid particles and creates a big food vacuole. And because it's eating solid food, it needs lysosomes, the cell's digestive stomach, to come in and break that material down. Pinocytosis takes in small, dissolved particles and fluids, creating a much smaller vesicle. Those dissolved substances absorb directly into the cytoplasm, so no digestive lysosomes are needed at all. Simple as that. Finally, Section 5, Summary. Let's look at the big picture. We have covered a ton of microscopic ground today, so let's make sure the absolute foundational rules of cell transport are crystal clear. Look at this ultimate showdown. For passive transport, you're always going downhill, from high to low concentration. That means it is a completely free ride, no ATP needed, whether that's osmosis or diffusion. But active transport, you are fighting the current. Moving low to high concentration will always cost the cell energy as ATP, period. Just like that hardworking sodium-potassium pump we just looked at. Which leaves me with this thought to wrap up our explainer. Right at this very second, millions of microscopic gates are swinging open and shut inside of you. Sodium-potassium pumps are firing, changing shape, burning ATP in billions of your neurons. Your cells are actively eating, drinking, constantly working. If all of this highly regulated, invisible border control is happening flawlessly without you even knowing it, what other unseen cellular miracles are keeping you alive today? Definitely something to think about. Thanks for joining me on this explainer.